And thank you to everyone who's joined the call. We're grateful for your support, and I'm excited to give you an update today. Can we have the next slide? Of course, we have a disclaimer slide just saying that I may make forward-looking statements, but Paul, I appreciate the background that you just gave, highlighting the accomplishments that we made over the last year. I know many of those watching this are interested to learn about the timing of trials and other advances of our program. So straight out of the gate, I want to give you a roadmap of the highlights that we're working on right now and will be working out over the next year. The first half of fiscal year 22 has been busy as we advance our clinical trials and establish research collaborations to expand our pipeline. Specific accomplishments include the recent signing of the clinical trial agreement with SUNY Upstate, the site for our PEACH clinical trial, and the announcement of the principal investigator, Dr. Chris Paolino. We're working very closely with SUNY to get this trial underway. In addition, we're working to finalize an agreement with the Clinical Research Organization, or CRO, to run the trial. We're working to complete manufacturing of the clinical material. In addition, we're working on efforts on both of the research collaborations and hope to kick off activities on both in short order. As we look to the second half of fiscal year 2022, or right after the new year, advances should really be moving along. It's going to be an exciting time. We expect to complete manufacturing of the clinical material, which will allow us to finalize and file the investigational new drug application, or IND, with the FDA. We're working on the IND right now, as a matter of fact. And once the IND is filed, we'll be able to start screening for subjects to enroll in the clinical trial. We expect to dose the first subject, as well as move to the cohorts of the study in the first half of next year. As to the research collaborations, these also will be well underway. We'll be screening libraries for new antiviral agents against different viruses with unmet medical need. As we look to the first half of fiscal year 2023, we will see the PEACH study continue to move through the cohorts until ultimately it's completed. At this point, we'll have the trial readout. We'll be planning to have a meeting with the FDA to discuss next steps. With respect to the research collaborations, we'll be identifying lead molecules to advance. So it's going to be a very busy year. This year, we have been very busy getting things ready to go for the clinical trial. A lot's going to start happening once we roll into the new year. Next slide, please. So having given you a roadmap of where we're going, I'll give you a background of the company, sort of a reminder for many of you, but ISLA Pharmaceuticals is a mid-clinical stage drug repurposing company focused on the rapid development of antiviral therapeutics for infectious diseases. As Paul mentioned, we had a very successful IPO about 6 or 7 months ago. It was heavily oversubscribed. We raised money, enabling us to move forward with our phase 2 clinical study, which we're really excited about. What we like about the program is that it's a very efficient strategy. It's a rapid approach to getting into the clinic, which we think is really important, a way to increase value in a very cost-effective way for shareholders and for patients ultimately. Our lead program, as you know, is in dengue. We have a phase 2 program, and we'll be rolling that out next year. Next slide, please. So we really like the drug repurposing strategy, and we're using it not only for ISLA 101, but it's going to play a critical role in our research collaborations with Monash and Griffith University. It's generally accepted that to develop a new drug, it takes about 10 to 15 years. It costs about $2 billion US. And that's not what we're doing. Because we're able to rely on molecules with a clinical history, moving straight into a phase 2 clinical trial, we get to start in phase 2. We get to eliminate essentially 2/3 of the drug development process and literally save hundreds of millions of dollars in the development costs. So it's a rapid way to move forward, and we like that very much. Next slide, please. So what makes ISLA 101 special? Why are we excited about this? This is a molecule that was initially developed about 20 years ago by J&J, and it was initially discovered and advanced as a potential cancer therapy, but it's never been approved anywhere in the world, despite being in 45 plus clinical trials. It's been in phase 1, 2, and some phase 3 clinical studies for cancer and other indications, but it's never been approved. But the benefit of that is that many regulatory agencies have signed off. They've let clinical trials begin. There are volumes of literature describing clinical results as well as preclinical results with this molecule. And so the safety of the molecule has been established and many people have signed off on it. It's just not been a very effective cancer therapy to date. However, we have fantastic data that it's a very good antiviral molecule. And as I'll discuss in a few slides, it has very good activity against a variety of viruses. So we're excited to repurpose that drug as a potential cancer therapy to an antiviral prophylactic molecule. We can literally rely on time, effort, lots of money spent by other people and move forward very quickly. Next slide, please. I mentioned we had a robust data package. So ISLA 101, we're really blessed that we've got a data package demonstrating that it has excellent activity against all 4 strains of dengue, as well as Zika, West Nile, and yellow fever. That's really important. These are all flaviviruses, mosquito-borne viruses, and it would be fantastic to have a pan-flavivirus molecule. We're keenly focused on dengue as a first indication, but there may be opportunities to expand, and I'll discuss what we think about that. Some of the data that we have cell culture samples where a variety of different viruses have been examined, all of those on this slide, in cell culture to see if ISLA 101 was effective, and it is. We have robust animal models and a Zika model, as well as a lethal model of a dengue infection, demonstrating that ISLA 101 is effective in animal models. These are the data that we took to the FDA in a pre-IND meeting and compiled a package that said, look at all of the clinical history that other people have developed, look at our preclinical data in flavivirus infection, and in particular dengue. Can we move forward with a phase 2 clinical trial? And the FDA said, yes, you can. So we like that. Next slide, please. I mentioned that numerous clinical studies have been performed around the world. This slide is showing you the number of clinical trials run in different geographies, showing you many regulatory agencies have let clinical trials proceed with this molecule. Again, highlighting the fact that the safety profile is quite excellent. Next slide. So this, I want to unpack this a little bit. This is a design of our phase 2 study. We call it the PEACH study. The reason we call it PEACH is because it really stands for a phase 2A randomized double-blind placebo-controlled study for the prophylactic examination of an antiviral in a dengue challenge model. And we decided that was a mouthful. So this is going to be PEACH going forward. This is an incredibly exquisite and exciting clinical trial. What we're doing is called a challenge model. And what this means is we enroll healthy individuals, we infect them with an attenuated strain or weakened strain of the dengue virus, and then we're able to track the infection because these folks will come into the hospital and do daily blood draws so we can check the viremia, we can check other symptoms associated with the disease. Now, a beautiful thing about this study is that we're doing this in conjunction with Walter Reed. This is the Army Medical Hospital in the United States. Walter Reed has already created this virus, and they've run control individuals through the study where they've just been looking at the viremia, looking at the viral load and other symptoms associated with the infection. And that study's done. We have a CRADA in place with Walter Reed, and what that does is it not only gives us virus so that we can use it in our clinical trial, but it's going to give us all of the data that they've already generated out of their control study. So in essence, our control arm is done. We now are going to enroll individuals and start pre-treating them with ISLA 101, then infect them with an attenuated strain of the dengue virus and monitor those folks. So it's a prophylactic study. But again, we're queued up and ready to go just as soon as we can get the IND open. We're excited to do this early next year, and it's going to be a very exciting study. Next slide, please. So where are we going to go from here? So we're going to file our IND. We'll start the PEACH study as we've described here on this slide. When we finish the PEACH study, we will plan to move forward with a phase 2/3 clinical trial in dengue and then seek NDA approval. There may be some optionality along the way. For instance, if we have an opportunity to pursue one of the other viruses, Zika, for instance, we could do that. Right now, we're keenly focused on dengue, and the plan would be to get an NDA approval for the dengue indication and then expand to these other indications. We could pursue later a study in Zika or chikungunya or West Nile or yellow fever, one of these other viruses, or potentially an emerging virus that we don't know about today. That's one of the beauties of the repurposing strategy is we can pivot quickly to attack emerging viruses if needed. But that would be our approach. Now, you'll see that I've highlighted Zika and chikungunya and dengue with a star with an asterisk beside them. And I did that because These are viruses for which you can receive a priority review voucher, which is an interesting opportunity that we have with ISLA 101. Next slide, please. So the FDA created an incentive program to encourage companies to develop drugs for underserved diseases. And there's a list of diseases for which you can get what's called a priority review voucher, and Zika and chikungunya and dengue are on that list. So what is priority review? When you file a new drug application with the FDA, it typically takes about a year to get your drug approved. If you get priority review, you can shave about 6 months off of that. You can imagine if you're developing a $1 billion drug and you're on the market half a year earlier, that could be $500 million of revenue, which is very important to pharmaceutical companies in general. Big Pharma really likes that idea. So the voucher program allows you to, if you check some boxes, and right now we're eligible and we do check all of those boxes, your drug is first approved in an indication that's on a list of diseases for which you can get a PRV, you get a voucher. And what that enables you to do is get priority review of a second program. So you get your drug approved in your first indication, you get a voucher that you can use for another program. And what companies do is they typically sell them to Big Pharma. And there's a market for these. The last 3 priority review vouchers, as you can see on this slide, sold for about $105 million US. The average of the last 10 is just over $100 million US, so it can be quite lucrative. We view this as a very interesting potential opportunity, sort of icing on the cake. Our goal is to get the drug approved, get it into patients. If this opportunity is there, we think it could be pretty attractive. So next slide, please. So the commercial opportunity for ISLA 101, there are many. We believe that there will be opportunities to sell the drug to travelers as a prophylactic, much like the malaria indication, malaria pills that people take. We believe that the military may be interested in this, and the fact that we're working with them right now on the clinical study is nice support for that. We think that there may be opportunities with national outbreaks. We know that India is having significant outbreaks right now with dengue, as is Latin America. There's a lot of problems there. I was at a meeting just 3 weeks ago, a partnering meeting in Latin America, a virtual partnering meeting. And the pharma companies that I spoke with there are really fascinated with what we're doing. They think it's just very important, and it's a very receptive audience to hear the story. So that was very nice. And again, we have the potential opportunity with the priority review voucher, which we think is, again, it would be nice if it's there when we get there. Next slide, please. Turning now to the research collaborations, just to highlight the Monash collaboration. We initially licensed ISLA 101 from Monash University, and they're phenomenal partners. We really have a great relationship with them. Professor David Jans at Monash has a system set up in his lab where he's able to study the interaction of viral proteins and host proteins and look for molecules that block that interaction to prevent the infection. That's exactly what he did when he discovered ISLA 101. So we want to repeat that. So the strategy that we'll employ with Monash is to again use the system in Professor Jan's lab to look for molecules that would prevent the interaction of a viral protein with a host protein, but a different virus, not a virus that we're working on right now. And that has yet to be determined. We're working on that right now. Next slide, please. The Griffith University collaboration is a little bit different. So at Griffith University, they have a very sensitive assay for measuring the weight of a protein. And in fact, it's so sensitive, you can measure the weight of a protein even if a small molecule attaches to it. And you can determine the small molecule by the change in the weight. It's very slick. So that tells you if a molecule binds to your protein of interest. It doesn't tell you anything about biology. So this is a two-step collaboration where with Professor Ron Quinn, we'll screen molecules. These are a library of compounds with clinical history. So we want to find a molecule that's been in the clinic before so that we can repurpose it, move to a phase 2 study. So we'll look for molecules that bind, working with Professor Quinn. Then we'll work with Professor Mahalingam, who has animal models of a variety of different virus infections. And so we'll take the binding molecules from Professor Quinn, we'll put them in the animal models, and again, look for relevant biology so that we can have lead molecules really coming out of 2 different programs. Next slide, please. So what does our pipeline look like? Well, we have ISLA 101, where we have our PEACH clinical trial that's going to be underway after the new year. And then we can expand that to other mosquito-borne viruses. So ISLA 101 really provides a platform for us, a pipeline and a pill, if you will, to pursue a variety of different viruses. We then will have programs yet to be determined with Monash and with Griffith University. Next slide, please. So I've mentioned a variety of collaborators throughout this presentation, and we have many, and we're grateful for them. Monash University and Griffith University, I've just mentioned them as the collaborators we'll be doing research with. We also are working with Compounds Australia. Compounds Australia is the group that houses the numerous libraries of small molecules. We're interested in the ones where the molecules have clinical history. So we'll be able to access those through our relationship with Compounds Australia. I mentioned the US Army. We have a CRADA in place with the US Army. They're wonderful partners. We're really excited about our clinical trial that's upcoming. Catalent is going to be making our clinical material, the clinical product, and we have an agreement in place with them. Finally, we're grateful to the National Cancer Institute. And you say, why the National Cancer Institute? Because you're an infectious disease company. The National Cancer Institute actually gave us the investigational new drug application for the formulation we're going to be using in our clinical trials. It saved us a tremendous amount of effort and time. So we're really grateful to them. Next slide, please. Just to highlight our team, we've got a phenomenal team. We've already talked about the board and the management. I do want to highlight our scientific advisory board, Professor Lee Farrell, Professor Stephen Thomas, and Dr. Simon Tucker. All three of these gentlemen are just fantastic scientists, world-class virologists, experienced in business development, in clinical trials, in drug development. Grateful for really a fantastic team here. Next slide, please. So key strengths of Island Pharmaceuticals, I think there are many. We're initially targeting dengue. This is a disease with 400 million infections a year. It's a significant unmet problem, unmet medical need. There's no therapies, there's very limited vaccine, there's a huge opportunity for us. And I think that we have a very good program to find a solution for that. The reason we get to move forward so quickly is because we have a very clever repurposing strategy where we can capitalize on efforts of third parties, money of third parties to move our program forward quickly. In the phase 2 ready asset, we've already had a pre-IND meeting with the FDA. They've given us the green light to move forward with our IND. So we're very confident and hopeful, obviously, that that's going to be approved. We think there's significant commercial upside. Again, ISLA 101 has the opportunity, at least based on the preclinical data, to serve a variety of these flavivirus infections. So we think that's a significant opportunity. Then we have established research collaborations where we can expand our pipeline, really turning into an antiviral drug development company. Finally, I can't say enough about our highly experienced team. This is a group of people that has really contributed. They've all worked hard to set the company up for a lot of success. So with that, I'm going to stop. We can go to the next slide for questions, but thank you again for your time.